The following video is a run-through of how you'd be performing DNA extraction for the 21 HPV diagnostic test kit. We will first perform the cell lysis for DNA extraction, followed by PCR amplification. First, vortex each of the samples to enhance the cells come off the brush. Each sample should take approximately 10 to 15 seconds. The DNA extraction process does not have to be done in the fume hood, but in a full-scale molecular lab, this process would be done in the fume hood, as illustrated in this video. Label all your microcentrifuge tube or screw cap tube with a permanent marker. Setting the pipette to 1 milliliter. Pipette 1 ml of cervical sample into a 1.5 ml microcentrifuge tube or screw cap tube. To obtain as much samples as possible, pipette several times before transferring the sample into the microcentrifuge tube. Please remember to use a different pipette tip for each sample. Please note that in order to perform this particular DNA extraction, you will need a centrifuge capable of performing up to 14,000 rotations per minute. Place the samples into the centrifuge. For those using screw cap tube, place a mark on the tube cap. The mark should be faced outwards during the centrifugation. For those using the microcentrifuge tube, the opening of the cap should be facing inwards. Please note that illustrated in this video is the screw cap tube. Centrifuge the samples at 14,000 rotations per minute for 5 minutes. After the centrifugation, discard the supernatants into a waste bucket, taking care not to discard the pellet as well. Gently tap on the side of the microcentrifuge tube to get rid of as much supernatants as possible. Set the pipette to 400 microliters. Make sure there are no crystals in the solution 1 bottle. After checking, add 400 microliters of solution 1 into each of the sample. Although it is preferred that you change the pipette tip after each addition, it is not necessary. After the addition of solution 1, vortex each of the samples thoroughly. This should take you approximately 5 to 6 seconds. After vortexing, boil the samples in a hot water bath of 95 degrees Celsius or above for 15 minutes. You could either use a water bath or a dry bath. In this case, a dry bath was used. After boiling, allow the tubes to sit at room temperature for 2 minutes. This is a bottle of solution 2. Set the pipette to 400 microliters. And then pipette 400 microliters of solution 2 into each of the samples. After the addition of solution 2, mix each of the sample gently using your hands. Let the sample sit at room temperature for 2 minutes. Centrifuge the samples again at 14,000 rotations per minute for 5 minutes. Again, the mark should be faced outside for screw cap tubes, and for the microcentrifuge tubes, the opening should be facing inwards.